The reason I wear sunglasses constantly is I have a degenerative eye condition, and I've actually thought about trying to develop a gene therapy for it and looked into it, but this is not a condition that's going to be responsive to gene therapies. If you think about medicine, you realize that there are a lot of diseases that are completely incurable or that there are a lot of treatment gaps. I work in non-viral gene therapy, which can fill some of those gaps, I believe. I work particularly in cancer therapies and vaccine development. One thing when you think about any kind of therapy is that there's always adverse events or side effects associated with it. And part of my research is to look into the minor side effects associated with non-viral gene therapies. These can actually be used to boost the efficacy of some therapies, but they should be controlled to increase the efficacy of other gene therapies. In the development of these gene therapies, I use a technique called in vivo electroporation. And this requires a strong collaboration between not only a biologist or medically oriented scientist such as myself, but engineers who develop the delivery technology. I think my area of research is cutting edge and will lead to a lot of different therapies. There's a lot open for development, both on the biology side and on the engineering side. I think with the intersection of these two areas, it would give a student a great basis for venturing out and developing their own new interesting ideas as they go forward in their careers.
So the mission of the lab is to do translational science. We focus on trying to come up with new therapeutic approaches for a variety of different diseases. And essentially what we look at is ways to make drugs more effective, as well as finding ways to be able to do gene therapy by coming up with new delivery modalities. A major area that we have focused on is coming up with cancer therapeutics. And if you think about a lot of times with cancer chemotherapeutics, they're very toxic and they don't actually work all that well. So we have looked at ways that we can make drugs more effective and reduce that toxicity when it comes to cancer therapies. And one way we've done that is to use electric fields to facilitate the entry of these drugs into cells. And that way we could greatly reduce the amount of the drug that you need to be effective. And it actually has become very effective. And this is something that has already been tested quite extensively in clinical trials and is actually approved for use in Europe, and hopefully in the next few years will be approved for use in the US. The key for any student is to find an area that they can get into that they can be passionate about. In our lab, it's good because there's a lot of different things that you can learn. There's a lot of techniques. There's a lot of different avenues that you could look at. It could be development of a protocol, it could be development of instrumentation, it could be development of how you combine these things and put them in and utilize them.
So students in my lab would be getting experience with a variety of techniques. One is the surgical techniques involved in manipulating and controlling biological tissues. Students would also learn to collect data from those tissues. In particular, we emphasize electrophysiological recordings. And so students would be measuring electrical signals that those tissues are generating. And then students would be learning computational tools and analyses to take that data that they've collected from their electrical recordings and build computer models or mathematical models to help them understand how those tissues are functioning in a quantitative manner. And then from those computational models or mathematical analyses, students would be learning how to then engineer or build devices, electronic circuits that can either mimic the behavior of those computer models or manipulate through the injection of electrical currents manipulate those tissues to do other things.
My name's Christina Niku, and I'm pursuing my PhD in biomedical engineering. I work in the lab of Dr. Chris Pisaglia in ocular neuroscience and neuroengineering. So one current project I'm working on is using a pressure transducer to measure the intraocular pressure inside the anterior chamber and recording that intraocular pressure along with other physiological factors that might be affecting it, such as body temperature, anesthetics, mydriatics, and environmental stressors. One key piece of advice I would have is to be eager in your studies to learn about the application of engineering in medicine.
In my view, engineering research of all kinds should apply scientific principles for the betterment of mankind. To this end, the biomedical engineering research mission of my lab is to make gene therapy a reality. The primary area of research in my lab has been the application of DC electric pulses to affect cells in such a way that they will uptake therapeutic DNA sequences. This generally does not cause harm to the cells and results ultimately in expression of the DNA or a therapeutic benefit. My lab has also been involved in developing the use of helium plasmas and corona charge that are a non-contact way of applying the same electric fields or same sort of electric fields to tissues by simply spraying charge in a gaseous stream on the surface of the tissue of interest. The students that have come out of my lab have gone on to be professors, postdoctoral scientists at national labs or other institutes. They have gone on to industry to fill various different types of positions. So workers or people from my laboratory have gone on to a multitude of different career paths. Go Bulls!